And now it's my pleasure to welcome the next speaker, who is Paul Brighton from Warwick, and we'll talk about natural killer cells in line with Nick's talk. Please.
Thank you. I'd like to thank Rita and Amelia for the invitation to talk here today. I'm going to talk about something that might be a bit different to some of the talks you've seen already and for the rest of the conference, because this story really stems from laboratory bench work, looking at a novel role of endometrial NK cells and how they interact with acute senescent decidual cells and the potential role that this has in implantation. So we know that different mammalian species employ different means to induce receptivity, and in the mouse this is driven by an estrogen surge. This is essential for receptivity, and it renders the endometrium receptive and also activates dormant blastocysts, and the net result is the implantation of several embryos and multiple pregnancies. In humans, however, multiple pregnancies aren't required, and this estrogen surge has been surpassed by spontaneous decidualization. And this is a multi-step pathway with distinct phases, and the myometrium will change from a cycling tissue into a receptive tissue through tissue remodeling. And this involves first a pro-inflammatory response, seen in red, and then a profound anti-inflammatory response, seen in blue. And we can recreate this in the laboratory by culturing endometrial stromal cells and then decidualizing them with cyclic AMP and MPA, which is a progesterone. And we can do this at various time points. And when we do this, excuse me, we see, I've used IL-33 here as an example. You can see at day 4, we see an increase in IL-33, which is a receptivity implantation cytokine, and that is lost by day 10. So we see a pro-inflammatory response followed by an anti-inflammatory response. And here I've used an example of SSD2, which is a soluble IL-33 decoy receptor, which will bind IL-33 and render it inactive. You can see this increases over the time scale, contributing to this anti-inflammatory response that we see here. And we know this secreted inflammatory response is important for receptivity, because if we take media that we've cultured from our cells and flush it through the uterus of the mouse, we then see an increase in a whole host of receptivity genes at day 4. You don't see any increases at day in unconditioned media, so that's media without cells present, untreated, or by day 8 when the anti-inflammatory response is kicked in. You do not see this. So we know that this timed pro-inflammatory response is important for receptivity, but it's also important for implantation, because we can take the same culture media from day 4, flush it through the uterus of a mouse, prior to embryo transfer. And when we do this, we see only at day 4 a big increase in the number of embryos that implant. At day 0 and at day 10, we do not see this implantation. So this leads us to question how the autoinflammatory response is generated. And to first understand this, you have to consider the cellular dynamics in the cycling endometrium. So at menstruation, the tissue will self-renew by the recruitment of endometrial stem cells. And as estrogen rises, you see massive proliferation. So this tissue will grow from about 1 millimeter to about 10 millimeters in about 14 days. And we also see, as progesterone increases, that this proliferation stops. We see cell cycle arrest, and we've characterized this to FOXO, PER2, and mTOR, some key cell fate decision makers in the endometrium. And when a cell will arrest like this, it has one of 2 options. So it can either become terminally differentiated, which are decidual cells, or they can become senescent. And it's this senescent population which forms the focus of this presentation today. So do we see senescence in our decidual cultures? Well, first of all, we need to consider what cellular senescence actually is. So these cells have stopped proliferating, but they remain metabolically active. And they will have a certain phenotype which is specific for acute cellular senescence. And the sort of characteristics that we see are concentration of senescence-associated beta-galactosidase, or SA-beta-gal, into lysosomes. So they operate at much lower pH. We also see an increased inflammatory response. So we see what's known as a SASP, a senescence-associated secretory phenotype. And we also see upregulation of tumor suppressor genes such as P53 and P16, which will inhibit proliferative genes. We also see other changes in proliferative genes, an altered chromatin landscape with HMGB2, which will prevent transcription of genes involved in proliferation. But we also see, I think my battery's dying, loss of nuclear protein, so lamin B1. We see an enlargement of the nucleus, and then we also see increase in heterochromatin foci. So these are small punctate staining within the nucleus that are involved in sequestering proliferative genes. So we know what senescent cells should look like. So when we looked in our cultures at some of these phenotypic characteristics, we saw an increase in cellular senescence as we decidualized. And you can see here we've stained for SA beta gal. And you can see by day 4 and by day 8, the blue represents SA beta gal. You see these pockets or islands of SA beta gal staining throughout the decidual time course. We can also measure SA beta gal quantitatively using a fluorescent assay. And again, you see an accumulation of SA beta gal as we decidualize our time points. But on its own, SA beta gal is not a good marker for cellular senescence. So we've looked at other markers as well, such as HMGB2. We see a loss of HMGB2 and upregulation of our tumor suppressor genes as well. Furthermore, we see an increase in these heterochromatin foci, which will rearrange the DNA to prevent proliferation of, prevent expression of proliferative genes. And you can see by Western blot or by ICC an upregulation of these punctate stains within the nucleus. So these images are matched for confocal settings and fluorescent settings. So we also see a loss of lamin B1. So it's disorganized and chaotic as we decidualize, again representing cellular senescence. And we see an increase in nuclear size. So we've quantitated the nucleus from several biopsies and several cultures here, and you can see that significant increase in nuclear size. So what we see are all the hallmarks of cellular senescence as we decidualize our cultures. But that's in tissues. Do we see something similar? Sorry, that's in cells. Do we see something similar in tissues? And we've collected samples throughout the menstrual cycle and measured SA beta gal in whole tissues. And you can see, compared to the proliferative phase, the secretory phase, you do see an increase in senescence as well. And when we look at some of our other markers via Western blot, comparing proliferative to secretory, again, in all cases, you see changes which suggest that what we are seeing in cells is also what happens in tissues. And we've quantitated P16, another senescence marker, via IHC from about 250 patients, and you can see an increase towards the late secretory phase as well. So what we see happening in cells is happening in tissues. So what does this acute decidual senescence actually do? To test this, we've got two compounds here, dasatinib, which will reduce senescent cells, and palbociclib, which will increase senescent cells. And these two compounds give us the ability to manipulate the level of senescence in our cells, and we can see what effect they have. So dasatinib will block tyrosine kinases, so these are constitutively active only in our senescent cells. They will block the phosphorylation of substrate, and the net result is a loss of pro-survival signals, so we can reduce the level of senescence in our cells. And we can see this here with our SA beta gal staining. Pre-treatment with dasatinib, you see a loss of senescence in our cells. So palbociclib, on the other hand, will increase cellular senescence. So most stressors will signal through these E2F genes to inhibit cell cycle progression. And P16 will activate cyclin-dependent kinases and increase cellular senescence. So now when we do our cultures, pre-treatment with palbociclib, you see an increase in cellular senescence, as we would hope. So we took these cultures and our compounds, and we looked at some of the inflammatory mediators that we think are associated with receptivity and implantation. And here I've shown IL-8. And you can see when we increase cellular senescence with palbociclib, shown in blue, you see an increase in the expression or the secretion of IL-8. And similarly, when we decrease cell senescence with dasatinib, shown in green here, we see a reduction in IL-8. And this was true for other implantation cytokines as well, such as IL-6, and less so for VEGF. So the take-home message is that the acute decisional senescence that we see in our culture will drive the pro-inflammatory signal during the window of implantation. So not all cells will senesce. There's also this second population here, and we've just done a little bit of characterization to show that some of the key decidual markers, such as prolactin and IGF-PP1, are unaffected by changes in senescence. And we also have some cytokines released by decidual cells as well, such as here I've shown IL-15. And pre-treatment with either dasatinib or palbociclib had no effect on IL-15 secretion. And IL-15 is important because it's one of the key immune cell recruiters. So we were wondering if immune cells were recruited to senescent cells to actually clear or eliminate them. And we all know that one major leukocyte population in the endometrium is our natural killer cells, identified via immunohistochemistry via CD56 antigen. And you can see both on the left and on the right, you see an increase in the level of UNK cells throughout the secretory cycle. And they almost triple from the early to the late secretory cycle. But you'll also notice as well that there's huge variability between patients at individual time points as well. So we were wondering if the acute senescent cells will organize their self-elimination by recruiting these immune cells. And to study this, we've isolated UNK cells from our tissue. So we can do this based on its CD56 antigen. And you can see here, when we stain these cultures for CD56, we get a pure population of uterine NK cells. And we can co-culture these with our stromal cells and see what effect they have on the level of senescence. And when we do this, you can see here on the left an increase in senescence as we'd expect when we decidualize. But this is now lost in the presence of NK cells, which suggests that they're targeting and killing only NK cells, only senescent cells, because you'll see in green, undifferentiated cells aren't affected at all. And there's an actual video we've got here. So this is a video where we've decidualized our stromal cells and then co-cultured with UNK cells. And you can see here's a UNK cell that it seems to attack and nibble the cells. And the net result is a loss of cell confluency. You can see these processes start to fade away. And more NKs come in. There's obviously a chemoattractant gradient here as more NKs join the party. And the senescent cell seems to die. So we don't actually see this when our cells aren't decidualized. So this only happens in decidual cells where we see cellular senescence. And if we time it right, we can see holes appear in our culture. So in the absence of decidualization, the absence of NK cells, cultures remain healthy. But when we co-culture with UNK cells and then stain for H&E, you see all these holes appear, which would suggest that the holes that you see here are also true of what we see in culture. So we did a little bit of mechanistic study as well, looking into how the UNKs will actually clear these senescent cells. And first of all, recognition. So many senescent cells will express these glycoproteins, which aren't expressed in non-senescent cells. And NK cells will express receptors which recognize these. NKG2D is one of the type of receptors. They will recognize these ligands, and it's one of the major recognition receptors to detect and eliminate transformed cells. And we can test this by using an antibody that targets specifically this anti-NKG2D receptor. And you can see now when we do our cultures, we see an increase in senescence that is lost in the presence of NK, but then reversed in the presence of an inhibitor of the receptor. So we know this receptor is involved in this mechanism. Furthermore, senescent cells will secrete senescence-associated phenotypes, so inflammatory cytokines, And we can eliminate these using molecular techniques such as siRNA. And here I've given an example of IL-15, which might be slightly misleading because it could be depleted from our decidual population. But when we do these experiments, we're comparing this one here with this one here, as we've knocked out IL-15, you see reversal of this senescence clearance. There is also several mechanisms that they could actually kill the senescent cells, and we've isolated it down to degranulation through perforin pores, the injection of cytotoxic proteases, and apoptosis. And we've tested this by inhibiting several stages of this pathway with 3,4-DCI and ZVAP-FMK. And again, when we inhibit this pathway with these two compounds, you see a reversal of the senescence killing. So this leaves us with a nice, simple working model that as cells decidualize, they have one of two fates, either decidual cells or senescent cells. And both these cell types will work in harmony to eliminate the senescent cells and perhaps curtail the pro-inflammatory signal that we see. So by their very nature, these UNK cells could potentially act as tissue homeostasis or tissue regulators. And we have this balance between clearance and senescence. So we would expect, therefore, that UNK cells would fluctuate between cycles. And sure enough, we've taken biopsies from successive cycles from the same patient. Here you can see sometimes NK cells remain low. We've also seen patients where NK cells remain high, but we've also seen fluctuations, so they can go from high to low, and they can also go from low to high. And we've done this in about 50 different patients, so each line here represents an individual patient, and you can see that as well as going up and down and staying the same, you can see that there's a huge variety of UNK scores that come through with our biopsies. And we've done this in more than 2 biopsies from the same patient, so we've done it in three or more, and you can see that they fluctuate mildly. And this will reflect their role in tissue homeostasis. So in summary, we've shown that an endogenous inflammatory signal has supplanted the estrogen surge in the human endometrium, and the inflammatory signal necessary for implantation is generated by cellular senescence. These decidual cells are then cleared by UNK cells via degranulation, cytotoxic degranulation, and this thus couples receptivity to tissue remodeling at implantation. And we now know that cellular homeostasis in the cycling endometrium is driven by the rate of stem cell activation, rate of proliferation, but also now the rate of UNK cell clearance. And it's really this implantation that may be governed by the temporal orchestration of all these various cell populations within the endometrium. And it may be important to get the timing and the balance right of all these various cell populations to have a successful implantation. So that leaves me to thank Professor Jan Brozens, who supervised this work. He couldn't be here today due to other commitments, but I've also got to acknowledge the work of Dr. Mariama, who was instrumental in all the culture work that was involved in this study, and also Dr. Catherine Fishwick, who tirelessly quantitated NKs from nearly 2,000 to 3,000 patients. And I should also acknowledge the support of Professor Shifon Quembe and the financial support from the Biomedical Research Unit and the continued support from the Tommy's charity.